Welcome everyone to today's webinar on leveraging soil microbiomes for resilient organic farms. I'm your host, Alice Formiga of eOrganic. This webinar will last between 45 minutes and an hour, probably close to an hour, and we will have another 30 minutes after that for questions. So today, I'm very pleased to welcome 4 members of a research team of a NIFA Organic Transitions funded project. We have 2 researchers from Cornell University, Elias Bloom and Ethan McNally, who are working in the lab of Dr. Claire Castile. And we also have 2 New York organic farmers who are part of the group of growers who are collaborating on the project. We have Leah Babbage of Turtle Tree Seed Initiative and Steve Christ of Fourfold Farm. So with that, we have a lot to cover today, so I am going to hand over the screen control to our first speaker, Elias Blooms.
Okay, hi everyone. Thank you so much for coming. I'm Eli Bloom, and today I'll be presenting some research that I'm titling Microbiomes Enhance Crop Resilience in Organic Farming Systems. I'll first introduce you to the breadth and scope of our microbiome project in New York State. And the first part of the project was mainly focused on doing a large study of how organic farming practices influence the microbiome. And some of that research will be presented today by Ethan, and he'll focus on telling you a little bit more about how composting influences the microbiome on organic farms. And we also recorded a webinar last year, and I've included that webinar link here. Go ahead and check out our previous webinar that's more specific on farming practices and the organic microbiome. The second and ongoing part of this project has focused on rapidly sequencing the soil microbiomes on organic farms and giving farmers decision support. So the idea there is that we can sequence the microbiome and we can relate it to other types of functions within the farming system, including pest suppression. Part of this project is also focused on giving farmers custom microbiome reports, and we'll be releasing some of those custom microbiome reports to about 85 farmers in the state of New York soon. Now, the focus of today is sort of the middle part of this project, which is a study that's looking into abiotic and biotic stress and how that influences the microbiome's capacity to help plants defend themselves from pest pressures. And this is particularly important as we think about how abiotic and biotic stress, like drought conditions and insect pest pressures, are going to be changing over time on organic farms. The origins of this study came a few years back when I started to learn about how plants are adapting to local microbiomes in natural ecosystems. What we know from these studies is that plants are recruiting different species of microbes, and then these microbes help plants become more fit in their environment. And one example of this is that different types of trees, they recruit or they bring in different types of microbes that help them to be more adapted to drought conditions. So we know that this type of adaptation is going on in nature, but we don't really know how it's playing out on organic farms. But what we do know about organic production is that crops are adapting to the conditions within those farming systems. And this research was led originally by, and probably even before this, by Kevin Murphy. And Kevin found back in 2007 that crops that are bred for organic farms typically perform better within organic farming systems than they do within conventional farms. And this includes enhanced yields of these varieties that have been bred for organic farms within organic farms. Now, Kevin thought that this was maybe mostly influenced by the crop's adaptation to nutrient conditions within organic farms, but we've been studying how potentially the crops are adapting to the microbiome of the farms, and that might be one reason that they are performing better within organic farming systems than elsewhere. And the third piece of information that's influenced this study is the idea that not only are crops adapting to organic farming systems, they're also adapting to local environments. And these local adaptations are helping plants become better adapted to the local abiotic conditions, such as drought conditions and pest pressures. and that this makes the seed better for farmers and benefits the farmers. And this has also influenced the regionalization of organic seed and the organic seed movement and the seed saving movement. And this is work that was led by the late John Navazio. Very important work. So our question was, how are these local organic microbiomes, how are they influencing the crop's capacity to defend itself from biotic stressors like insect pest pressures? So are plants that are grown in their microbiomes better defended from biotic stress? And then how is that going to change, or is it going to be sustained when we also have abiotic stress, like drought conditions? So like Alice mentioned before, we've been working with 7 organic seed producer stakeholders in the state of New York. These are the farms that we've been working with, and this includes Leah and Stephen. And I've been going out to these farms a couple of times over the last couple of years to collect soil samples from each one of the farms. We take those soil samples and we extract the microbiomes to use in our experiments. And I've also been working with Stephen and Leah and the other farmers to collect seeds that we use in the experiments. So then what we do is we take the seeds. In this case, we've been working with 2 of the seeds so far. One is kale and another one is sai. We've been growing those kale and sai in the microbiome of the farm that they came from. And then we've also been growing those kale and sai plants in the microbiomes of all the other farms that we're working with. And then to simulate that biotic stress, so how is the plant responding to insect pest pressures when it's being grown in its microbiome versus the microbiome of other farms, we take aphids and we put them on the plants and we evaluate how many aphids are produced on these plants when they're grown in their microbiome and in the microbiome of other farms. So I'll walk you through some of the results from the study here. On the x-axis, what we have is the different microbiomes. So we have a control microbiome, which is a no microbiome control. We also have the microbiome from which the kale seeds came from. And then on the second plot over here, we have the control again. And then we have the comparison to the sai seeds grown on the sai farm, or with the microbiome of the sai farm. So what we can see is that when you grow plants in the microbiome where the seeds are produced, they have substantially increased pest suppression. So they have fewer aphids that are reproduced on those plants, which indicates that the microbiomes of the farms where the seeds are being produced are enhancing pest suppression. Now, if we scale that out and we look at all of the other farms that we're working with, you'll see that a pattern emerges here. And if you look on the left here under kale, you'll see that several of the farms here with the red brackets, and if you look at sai here, again, with the red brackets, you will see that many of these farms also have similar pest suppressive capacity to the farm where the seed is being produced. So what we're able to say is that the New York seeds that are being produced here in New York also are able to promote regional pest suppression, in that if we use the seeds on the other farms in the other microbiomes, it appears that we also gain those pest suppression benefits from the microbiome. And then one final observation that you might see here is that some of the farms, such as farm 3 and 4 here, these farms have even lower pest numbers when the sai seed is being grown in their microbiome than the farm that the sai has been originally produced on. So these farms might be supercharging pest suppression. And so looking into why some of these farms are able to help plants defend themselves even more from pests is an open question that we're looking into. So the last part of this study, or the last part of this experiment looking into the biotic stress, is how does that change when we also introduce drought conditions? So how are plants able to defend themselves from these pests under droughts? So again, we ran this experiment, and now we have the plants and they're under droughted conditions. And what you'll see is that the kale plant is able to defend itself from pests as compared to the control under droughted conditions. But with this sai, we actually see that the pest levels are very similar to the control. And so what this is saying to us is we think that it's possible across different plant species and different breeding strategies to push farms to be able to adapt to these droughted conditions and be able to defend themselves from insect pests. And another observation that we've made from our study is that, again, some of these farms, such as farms 2, 3, and 4, have similar pest suppression levels to the farm where the seed is being produced under droughted conditions. But these farms also appear to be highly functional for multiple plant species, 2, 3, and 4. Their microbiomes are also very functional. And so what we think is that potentially some farms have functions that extend to multiple different plant species in terms of their microbiomes. So at the end of this experiment, we took the plants and we shook off the roots from the soil. And we took the soil and we extracted the microbiome and we looked at what was going on in the microbiome. And I'm just going to summarize some of the results for you. First, one of the big take homes was that farms have unique microbiomes. And these microbiomes, even though they're different, they're also equally functional. So they're able to promote pest suppression regardless of what the composition of those microbiomes are. We did find that just like in natural systems, these different crops are attracting different microbes, but it kind of varied for the crops. So the crops that were better able to change their microbiomes under drought conditions, that kale plant was actually better able to then defend itself, whereas Sai-Sai's microbiome kind of changed in a different way under drought conditions. So we think that this change under drought conditions is possibly one of the reasons that kale is kind of able to defend itself under these droughted conditions. So what are some big take-home messages here? I wanted to give some kind of practical understanding of why we think we found the results that we have. And so we've not only have we run these experiments, but we've been working with the farmers to better understand some of the production characteristics. And so what we know about the production characteristics for this kale variety is that it's been grown in field conditions. It's been previously exposed to drought conditions, and it hasn't historically been irrigated. And so what we think could be going on there is, since the seed has been experiencing drought conditions and doesn't have prior irrigation, it's able to defend itself from insect pests under those droughted conditions when we grow it in the laboratory conditions, and it's able to recruit those microbes that it needs to defend for itself. Whereas we kind of picked Sai-Sai because it's been grown typically in the greenhouse and it is not experiencing droughted conditions and it's been irrigated. And so it kind of makes sense that this plant is able to defend itself when we give it all of the water that it needs, but under droughted conditions, it's perhaps not as able to recruit microbiomes that it needs to use to defend itself, at least not in all situations. And so one of the things that I'm thinking about, and I'm not the first person to promote this, is that a certain amount of stress when we're breeding plants might be useful to be able to have plants that are adaptable across lots of farms to environmental conditions, including drought and pest pressures. The second area that I wanted to highlight links back to my ideas about decision support. And I'm just sharing this here. This is from Leah's latest catalog at Turtle Tree Seed. And they're already thinking about this by telling the purchasers of seed where the seeds are produced and the production characteristics of those farms. So in the future, we might think more about what are the microbiomes of the farms where the seeds are being produced, and then what's the microbiome of your farm? And then how can we link the microbiome between those two farms to be able to provide seeds that are going to be best adapted to your local microbiome and be able to defend themselves from pests under future pressures like droughted conditions. And the final area that I wanted to highlight is the idea that some of those farms are supercharging pest suppression. Some of them have super high levels of pest suppression, sometimes even more high levels of pest suppression than the farms where the seed is being produced. And so the idea there is that potentially we can build up some regional collaboration and seed production networks focused on helping plants adapt to microbiomes that help them to be more resilient to these types of pest pressures and abiotic conditions like drought, and use those regional collaborations to promote seed that is adapted to those conditions in the future. With that, I will hand it over to the next speaker, but I wanted to acknowledge and thank all of the different people that have worked on this project, our funding sources, and of course, a special thanks to all of the farmers that have been working with us over the last couple of years on this project. So thank you very much, and I'll hand it back to Alice.
Yeah, thank you, everybody. I am representing Fourfold Farm, which is the production wing of the Hudson Valley Seed Company. So we are kind of a separate entity on paper and for organic certification, but we supply exclusively to the Hudson Valley Seed Company. And we grow a large biodiverse amount of seed and tubers and sunchokes and alliums, et cetera. And here I listed one of the seed company's 4 pillars. Mission statements is we know seeds because we grow seeds. And I particularly am very proud of the company that decided that they needed a working organic seed production farm as opposed to just being a redistributor. Keeps me employed and lets us do what we love. Just a beautiful picture of some perennial echinacea here. A little about me, this kind of slightly American Gothic autophylle polenta corn picture is this is from last year where we grew close to 800 pounds of polenta corn. I am fascinated by the miracle of seeds and the stories that they tell. I'm in love with organic farming process and the medium of soil, building a farm from headspace to vertical fourth dimensional reality is the art form I'm the most passionate about. I'm deeply interested in achieving resiliency and bounty within climate chaos, where patterns and seasonal variability are very hard to predict. And I love seeing soil improve with dedicated cover cropping and different methods. And the stories that seeds tell, this autophylle is an Italian polenta corn, but it most likely came from right here in New York from the Lenape native tribe and went to Italy and was selected for 8 rows of corn. And getting it back here has been interesting in selecting it further. Our philosophy on the farm is to grow the highest quality organic open pollinated seeds, tubers, alliums, and seedlings for the Hudson Valley Seed Co. in adherence with a sustainable and ecologically ethical model, minimum inputs, controls, and a high fertility budget, good compost, multiple field strategies, sometimes no-till, sometimes reduced tillage, cover cropping pathways, living pathways. We believe that soil and seed are everything. With a huge range of plant type each year, we are a very biodiverse farm. It's a very, from watermelons to nicotiana, the size and the needs of the seed are huge, hugely variable. And soil is the common denominator, which is a big message I'd like to get across here today. The blank canvas is a microbiome that thrives within organic systems. This is a belief of me and our wonderful farmers. And this picture is a photo of what will be 9,000 bed feet of dahlia tubers. And it's one of my favorite times of the year is seeing this blank canvas come from the headspace and the spreadsheet into reality and then go vertical. That's one of the biggest joys I get in life, really. And I couldn't do it without our amazing crew. It's not a very large crew, and we had to do some additional part-time people not mentioned, but I felt quickly to mention our field manager, Kendall, trials and specialty gardens coordinator, Katie, greenhouse coordinator, Bill, and some other wonderful seasoned farmers, Kenny and Ellie. They are bound by our shared philosophy and mission, and teamwork makes the dream work. We have, there's like 7 of us at max at the height of season, which is a lot for 15 plus acres. So really appreciate them. Just quickly here, everything does start in our greenhouse where we have maximum controls, but they can't stay here. That's obvious. The seedlings go right to the customer, but everything for our production farm starts very well controlled, and then it goes right into our soil systems. At the end of the year, after it's had that extended time in the soil, we convert our greenhouse to a barn where the fruits of our labor dry down. But the field, the medium between nursery and processing the soil is where the real work happens. A little bit about seed saving. I won't take too much time on this, but just for those that aren't maybe very well informed, or I hope it's not too pedantic, but there are 2 types of seed saving, and it's an elemental process. Dry seed processing uses air and wind to separate seed from chaff. much like our ancestors did, but with some modern tools. Winnow wizard, air columns, not pictured. Dry seed harvesting represent the ultimate expression of a plant's life cycle. What is otherwise dead and dying to some is our product. We see beauty in that final stage because it really is we go from A to B to C, back to A again. It's a beautiful cycle. And we have some Japanese indigo on the top there and some straw flower, very, very small seed. And then there's wet seed processing. This is a lemon drop, I believe, or Ho Chi Minh hot pepper. Using water and sometimes fermentation to allow heavy and viable seeds to sink so that immature seed and unwanted plant matter can be poured off. Unused material is added to the compost pile and slowly turns into a resource we can use again. Of course, if there's no disease. Other items like edible winter squash are donated to food pantries and animal shelters. The fermentation process is really so that like something like a tomato that has a bit of a mucilage around the seed can actually burn off, allowing the heavy seed to sink and the light material to be poured off so you can get very clean, pristine finished products. And this was, I believe, a black futsu squash from last year. And again, we want those seeds to have a high germination rate. So getting them clean and winnowed is the mission. But a little bit more about like our soil practices and to tie it back into this discussion today. For seed growing, we have extended times in the soil. It is both our challenge and our gift because, for instance, there's something called the blowing up in the kiln effect in the Northeast is what I call it, when you get a perfect seed crop to right at the point where the seed pods are matured and they're ready to harvest, but then it rains for 10 days and you're fighting against time and you can lose seed to wind and water very quickly. This is French breakfast radish on the left at one stage and from a different angle about 3 weeks later. And that turned out to be really good for us, but it was the first time we ever grew French breakfast radish. So we did see some insect pressures, and it was not as easy as some things that we've done over and over again. Seed crops that make it through the challenge are almost always more resilient year to year. We believe this has everything to do with adaptation, directly linked to the microbiome and seed memory. That's a slightly pseudoscientific term, but that's how I call it in the field. The more we grow a crop, the better it becomes, is the long and short of it. Just a word on, this is a great word, lots of S's. Senescence is the aging product of a plant. So it is necessary for the production of seed. Overwintering brassicas are a prime example of a crop having extended time in the soil. And these pictured here are on the verge of senescence. It's a kale and then a wild broccoli rabe in the foreground. And we are trying to harvest these things before the seeds shatter or the pods split open and drop to the ground. So in the Northeast with very wet climates, sometimes we have to do that earlier than we would if it was a perfect climate, like in Northern California, where some of the Navazio books are read. I'd have to read those books and alter my strategy a little bit and bring them into the greenhouse early to let them cure up. And I've found that if you do it at the right time, a green senesced plant can mature into viable high germ testing seed. You just can't do it too early. Something to say about organic matter, something that we believe helps the microbiome and helps the tilth of our soil and is part of our diverse strategy to reduce tillage. In as many cases as possible, we use a lot of mulch, compost cover cropping, rotation, etc. In this picture, we have a Cherokee purple tomato about to go into some beautiful rye straw. Compost is down below. There was a cover crop the year before. Getting the timing right is sometimes hard, but this purple Cherokee was the first thing dead in our fields that year, despite all that, but it was the first year we had ever grown that seed. It came from another source in the southeast of the United States. To the right of the chamomile that you can see on the right, there was a tomato called Goldie, which I've grown every other year for the past 10. And I was harvesting tomatoes in October. So that was a really cool thing to see and kind of juxtapose here. Organic matter continued. I love incorporating cover crop and field plant material back into the soil, as long as it's not Phytophthora or something infested, where no-till practices are not possible. A lot of cases we cannot be a no-till farm because the bed feed is just so much and our workforce so small. So, this is I believe an old cornfield that we had intersowed with some cover at the last cultivation. And then all of that stuff gets flail mowed and brought back into the soil. We've seen an incredible increase in soil tilth year to year. Reduced tillage, living pathways, minimum additives and controls, just some more bullets on like our philosophy. But this was kind of a happy accident, a chamomile seed crop that was very hard to gather seed from the year prior because of a very rainy year. Couldn't get my quotas for seed, but then I was able to let that come back, use it as an insect repellent in my allium fields, and it was the best allium yield we had ever had. So there was some predatory insects coming in and helping us with the allium insect pressure, and that was just a really cool thing. Skip these. We are like invested in continuing our work and finding other ways of supporting the microbiome. We were awarded a biochar grant this year, which we're going to be putting down for extended period of time. We've also been doing some experiments with localized grain. Don Lewis is a miller in our area, and we've been doing grain trials of some ancient grains, einkorn and emmer, and selecting the ones that year to year outperform the others and working with them. And we've seen a great improvement in the seed that we save from year to year and regrow in the same location for the grains, which is very interesting. We're also investing a little bit, kind of like what was talked about for Turtle Tree's catalog, in identifying where things come. But in this case, we're trying to diversify local ecotypes or endangered pollinator-friendly varieties for the Northeast, bringing in potential pollinators, and the link to healthier, more living ecosystems, including the microbiome, is as above, so below, where the insect life is thriving and pollinating and the soil systems are healthy. It's a little hermetic anecdote. Just the interconnection between native species and the native soils is something that I'm really fascinated in through this project it's called Pollinate HV, where we're just showcasing about 15 different native flower and endangered subspecies in the Northeast. Picture of the seed packs. Trying to be careful on time here. Climate resiliency is a big interest for us. We go from deluge to drought in the Northeast. The pendulum is swinging wildly with a trend towards a warmer, wetter climate. This is a year of deluge where our lettuce plants, we had not tied them up yet, and they are all flopping over. I thought I had time, but I didn't, and it was awful. And we also saw Phytophthora in our winter squash, and it was a really extreme year for climate chaos. 10 inches of rain in 2 weeks. So learning from our mistakes, something I call farm judo, and the practice of being prepared for everything, drought and deluge. Even if you've been farming for 2 decades, you've still only done it 20 times. It's a really long learning curve. And just an example, this is a year of drought where we had a lot of thirsty insects and incredible insect pressure, but we tied our lettuce up early. This particular lettuce I had grown a number of times and it did quite well, bolted early, produced a lot of beautiful seed, but was tied up nonetheless very early, just in case that hurricane came through before I had done my tying. This is just kind of a little mushy, but we do it for beauty. We do it, these things are gifts from the soil and our air, water, and pollinators, gifts from ourselves to ourselves. And we do it because it's food, and the ability to share food with others, to cultivate true excellence, takes a well-nurtured soil system. And we are fully invested and proud to be a part of that and proud to share that with others. Wrap it up. I think I'm okay on time. A couple more slides here. This is just a really cool image that shows that it does take time, but the power of soil is quite a thing. This is a Palestinian gourd called yaktine, growing and climbing over some tithonia. So you have a plant that originates from Palestine and one from Mexico, thriving in upstate New York. They almost all died in the field, but I spot sowed them, and we're still able to get a seed crop out of this very kind of removed gourd. And that was a really incredible thing to see. And I imagine when we do grow it again, that we will see an even punchier, more robust seed. An ode to dirt and all the potential it holds. Just a great picture of what I see as like completely beautiful. The farm often joke that we would love to just lay down in the soil when we're tired. We just want to be inside of it. And this to me has a lot of worm castings, a lot of root material. It just seems healthy. So it's something I take great pleasure in, is just dirt, because it brings us all these beautiful things like balloon vine tunnels, etc., and things that we can create and get better at year to year. So thank you all. That's what I have to say.
Hi, everybody. I'm Ethan. I'm in the same lab as Eli and Claire Castile's lab at Cornell. And I've been a research technician here for the last year and a half, and I'll be starting as a PhD student next fall. I'm somewhat new to agricultural systems. I have more of a background in microbiology, but it's been really great to work in for this last year and a half. So a little background, our lab did some work in 2020, just looking at the differences overall between the effects of conventionally managed and organically managed microbiomes on plant resistance to insects. And this was in California with tomatoes, but they found a pretty strong effect that organically managed microbiomes promoted more resistance to pest than the conventionally managed counterparts, which was really cool to see. And we're really curious with our current work to look into exactly which practices might be driving this. And one of the first steps that we took to do this was a big project that Eli had a big hand in, which was a survey of New York organic farmers on their beliefs as to which practices might be promoting plant resistance to insects through the soil microbiome. And in addition to this survey data on beliefs of what practices could be driving this, we also collected soil samples to actually sample those microbiomes on their farms and look at it compared to the practices and crops that they were using. And I'll get a little into this data later. But first, just looking at those beliefs, of the practices that we surveyed, the three that were seen as most important by New York organic farmers were composting, cover cropping, and crop diversity in fields. Zoe, a master's student in our lab, was able to present a bit on cover cropping last year, but my work in the lab has mostly been with compost. And starting out with this question was somewhat difficult, just given the complexity of compost. There's a huge diversity in terms of what goes into compost, how composts are managed, whether it's with huge thermophilic systems, with these windrows and tractors managed by worms, or smaller scale operations on the farm. And then compost can be applied in different quantities and in different methods at different times of year. So it seems like there could be some diversity introduced here. Additionally, when applying compost, you're clearly not only thinking about insect resistance. There's lots of benefits in terms of fertilization, improving soil texture, just as a way to manage organic wastes and reduce carbon emissions. But I really wanted to look into the effect on pests specifically. And just in terms of the work that's been done on this so far, it seems to overall agree with the impressions of New York farmers that compost promotes defenses, which was found in 17 studies. There were also a couple studies finding no effect of compost on insects, and one study finding that it actually reduced defenses, which these were in a variety of systems, some in the field, some in the greenhouse. So we wanted to look at our own data and see what we found. So back to that same set of New York organic farms, there were 10 farmers who submitted samples from both composted and uncomposted plots on their farms that were identically managed in all the other ways that we surveyed. And I was able to look at those microbiomes and the insect suppression of those microbiomes through a model that Eli helped develop, and saw really inconsistent effects. So basically what you're looking at here is on the x-axis, we have the same farm, a plot, whether it had no compost or was amended with compost. And then the y-axis is the predicted effect of that microbiome on insects. And in some farms, it seemed that insects would perform much better in the microbiome with compost. In some, it seemed that there was an insect suppressive effect. And there's a lot of things that we were thinking could be causing this inconsistency. These farms were growing different cropping systems. We didn't survey on the compost that they were applying, just that they were applying compost. So we're unsure about the composition here. They have different soil microbiomes to start out and different soil types. But it just, in microbiology, I'm really interested in this question of could the soil microbiome that's already present at a farm not only change the impact of the plants that are currently growing there, but how the soil responds to changes in management. So the way that we set up this experiment was collecting soils at 4 New York farms and extracting the microbiomes from those soils. So we take these soil samples make a slurry of that in a nutrient solution, and then apply that to different soils where we grew kale plants. We chose kale just since it's a really common organic crop and it's easier to work with in lab with aphids. And in addition to growing kale in these different microbiomes, we also grew them with or without standardized commercial compost. We use Cayuga compost here in Ithaca, which is managed with windrows and is composed of wood mulch and food scraps. And then after these kale plants were about 4 weeks old, we applied aphids and then measured their performance by counting the progeny of how many babies they had while on those leaves. And first of all, I just want to go over the plants that were grown in no microbiome. So basically, these are kale plants in sterile soil where we either applied compost or didn't apply compost. And on the y-axis here, this is the actual progeny of the aphids, of how well those aphids performed in each treatment. And we saw really no effect of compost. There was about the same aphid performance in both the uncomposted and composted treatments, which was kind of interesting. We were expecting to see somewhat of a suppressive effect given the literature, but it was really curious given what we saw in the soil microbiomes. So in these 4 farm soil microbiomes that we tested, compost did actually have effect in 3 out of 4 of them, and it was not the same effect in every microbiome. So this is the same on the y-axis here of aphid performance. And then in this red farm from Hudson Valley, you can see that aphids performed much worse when compost was applied. So it seemed that compost was having an insect suppressive effect in this microbiome. But in this blue farm here from the Finger Lakes, aphids did better when compost was applied. So completely the opposite effect of the same compost, just given the microbiome that was present in the soil when it was applied. Moreover, we did this same experiment 2 years in a row. And while the effect of the microbiomes on most farms was consistent, we had 1 out of the 4 that the effect of compost changed from 2024 to 2025, which there was a couple practices that changed on all of these farms between these 2 years. So we're not exactly sure what's causing this change, but it's interesting to see that the microbiome is malleable and the response to compost can change over time. And in addition to looking at the effect on aphid resistance, we wanted to look at how compost is impacting the microbiome itself. And the way we did that was taking the treatments from 2 of these farms where we saw the opposite effect of compost on aphids, growing those plants with and without compost, and then sequencing the DNA from the soils to basically read which bacteria and fungi are present there. And one of the main ways of looking at this data is just looking at the diversity of the community in the soil. So here on the y-axis is the diversity of the bacteria that were present. And on the x-axis, this is those same 2 farms with and without compost. And we found that compost consistently increased the diversity of the soils, which is consistent with what people are finding in literature of compost, but really interesting given that soil diversity is often upheld as a way to increase pest resistance. And we're seeing that the effect of compost is going beyond that. There's something that's going on not simply due to soil diversity, which it seems to consistently increase, which is causing this resistance to aphids. So overall, moving forward, the main conclusions we're taking away from this is that it does seem to be true that compost can support resistance to insects, but it's dependent on the soil microbiome on the farm where it's applied. It's also encouraging to see that, at least in some cases, that's not set in stone. Changes in management or changes over time can cause a farm to respond differently to compost. And we might be able to get a better understanding of this by using our decision support tool, where if we could take a sample of soil, sequence the microbiome in it, look at the DNA, we could maybe predict how that farm would respond to compost or other management changes, and if they could be beneficial. Also, in the future, we're hoping to look at more different kinds of composts, see if maybe some of the effects are more consistent. There's some really encouraging research out there on vermicomposts, and this is definitely still a work in progress. And then lastly, it's still really unclear how this effect is happening. With that research in 2020, we were seeing changes in plant defensive hormones that were causing changes in aphid performance. But how this effect of compost is occurring in the plant, we still don't know. So that'll involve some more molecular biology, and getting an understanding of that could lead to more informed organic management going forward. So just really practically thinking about how to apply this on the farm, I think given that we saw these differences in compost, it depends on which farm you're at, and it will make a lot of sense to do some experimentation, see how compost is affecting things, and work from there. I just want to thank the same people that Eli was just thanking, but this is our research group here, and this has been a really great stuff to work on. So I'll hand it over to Leah.
I'm just so grateful for everything you guys have just offered. It's been just absolutely wonderful to hear. And I have to say, so many of the things, Stephen, that you were saying, I was just nodding my head along with you because, yep, those are common experiences for us too. So I'm just going to tell you a little bit about turtle tree seed. And I'm so glad, Stephen, that you really gave a good background on seed growing, because what I'm presenting is a little bit, those are all things that we experience as well. But turtle tree is a pretty, unique entity. So I'll just give a little bit of that background of our history. It was originally, the idea for turtle tree was started in 1991. Beth and Nathan Coram were listening to their fellow farmers in the biodynamic and organic seed movements, biodynamic farming movement. And at that time, there was no biodynamic seed being produced in the US. If you're interested in what biodynamics is, please feel free to go and find that information. It is available. And also, I think very little organic seed production was happening at that time, possibly just some starting here and there. So they went and trained in Switzerland and came back and started a seed company, which traveled around for a few seasons trying to find their home. And eventually they found their name, and then they found their home in 1998 in Camp Hill Village in Copake, New York. So Camp Hill Village was started in 1961 as an inclusive community to support the needs of adults with developmental challenges. And when Beth and Nathan moved Turtle Tree there in 1998, it was a thriving community. There were about 22 houses with shared living for people with and without developmental challenges, which there still are. And there were 4 land areas where people worked. And those were the dairy farm, the vegetable garden, the estate and forestry, and the newly created healing plant garden, which grew flowers and herbs, both culinary and medicinal herbs, for use in the village and beyond. And so inviting the seed company to come, or seed work to come to the village, seemed like it was really completing this picture of a whole farm organism. And so that was a really wonderful step. Now, pretty much in contrast to just about every other seed company that I can think of, and certainly seed company, seed grower, we'll just say, which is not, of course, the same as seed companies, as Stephen will very well know. Most seed companies, of course, are just pass-throughs for seed which they purchase. And a few of us, Stephen's, the Hudson Valley Seed Company, and some other awesome companies in the West Coast, and a few others across the country, are actually growing our own seed that we sell, and have also a network of farmers who we work with who grow seeds and are transparent about where they're actually coming from. So we have this very, very different model where instead of having a few hands to work a lot of acres, we have a lot of hands to work a few acres. We work with folks with developmental challenges. We work with people from all over the world who come for a year, usually without any previous agricultural knowledge. They come to work with adults with developmental challenges, and they are placed with us as part of their volunteer year. And they may not or may not know which end of the carrot is supposed to go in the ground. So that's always an exciting and interesting challenge. You can flip this slide. So over the years, the folks with developmental challenges who found their calling in the seed work, along with some of us who have also found our calling in the seed work, have become the teachers of these young people who learn for a year and then go out into the world now knowing a little bit about seed growing. And that's been a really rewarding thing, as well as the seeds that come through our hands. So this is Andrea, who's had been a longtime seed person, and on the right, and Joseph on the left. That was his first year with us when he was just learning. Joseph is from Kenya, and he's now become one of our main seed farmers and a team leader on the farm. So at the beginning, we started out with about 40 varieties of vegetables and flowers when Beth and Nathan came to the village. And over the years, that was so in 1998. And over the years, we've built up the catalog of about 400 varieties. We don't do tubers, and we don't do allium starts. We do allium seeds, but otherwise we have a pretty reasonable catalog. And we are continuing to find out that some of the varieties we carry are unique to us. And recently, for instance, we just found out from a company or a farm in California that we were the only source of a certain melon that they had been relying on for years. And so we figured that we better start growing that out more regularly and with more intentionality, seeing that we're the only ones left with it, carrying it. We had been a little bit, we had a good seed supply, and we were like, all right, we don't need to grow this out for a couple of years, but suddenly it seems a little more urgent. So and that was totally unknown to us, that no one else was growing that seed. So it's we take that responsibility for the seed varieties that we're carrying very, very seriously. We also have young volunteers who come through, and also we have school groups who come through, and day volunteers who come for a short time or a long time. Sometimes there's someone who is retired in the area who would like to just volunteer on a Tuesday, or sometimes there are people who, through their work, have a volunteer requirement for their job that they come to us for one day, and that's lovely as well as the people who are there continuously. But we are always in training mode. This is Rukin and Ollie chatting about the Galanzoga in the squash plants and pepper plants. I hope they're also pulling some out, but I'm not sure. So with each season, there's new challenges, and we learn and adapt and grow, and we see the necessity of continually growing out varieties. We notice that new pests are, growing and appearing. New diseases are moving up from the south. We notice as we talk to farmers further south, things that are very common there are becoming more common here. And there are also these consistent things for us, these that we are, adding our compost that's been treated with biodynamic compost preparations to the soil every year, that we're applying our biodynamic field sprays to the soil and crops every year. And along with our changing conditions and the constant work of our biodynamic practices, we're also selecting plants that can deal with a variety of care from people who may or may not know what plants are, or who may pull up all the plants in the bed and leave the weeds. And then we have to replant all the plants. And if they can tolerate that, they can pretty much tolerate anything, especially when it happens more than once in a season. So the varieties that we carry have become like our children and our friends. They've grown with us, and we've watched their steady improvement and occasional shenanigans. And they've become rooted in the context of this valley, both agricultural and social. And this is an ongoing project. After adapting to very dry springs followed by wet summers, we've now had the wettest May in many years. My goodness, has been wet, and we're expecting new challenges to arise as things continue to change. So I just want to add that the built-up observation of these varieties as they grow and experience our land, and as the land experiences them, has just built up this bank of experience in the land and the seeds and the people who care for them. And this is part of our team. We have a very large team, but this is some of them.
A great photo. Eli, do you want to say any concluding words here for the webinar, or otherwise we can move on to our question and answer session?
I think I'm happy to move on to the question and answer session. I want to thank Leah and Stephen, and it's all very much tied together, the social aspects and the ecological aspects of producing seed that's adapted to these regional conditions, and the observations that they've made about deluge and drought and how that can change within the same year is something that's also been impressed on me, having worked with farmers in this state too. And so it's very interesting and continues to be interesting to kind of learn and think about how plants are responding to lots of different types of environmental conditions, but also the social context that Leah so eloquently described for how we're able to have these seeds that are so well adapted to this region. So it's just very interesting to be on just a very small microbiome aspect of that, and so nice to hear about how it's very holistic and it's everyone is coming together to study this. So I don't know if we want to continue along those lines or not, but I just wanted to thank everyone again, and I think happy to open it up to questions. And
Yeah, okay, great. That's great. We have we have another half an hour or so in case anybody has any questions for any of the presenters or anything about the research or about the farms. I just wanted to let anybody know who missed the beginning of the webinar that we did record this, and the recording will be available both on the eOrganic website. You just Google webinars by eOrganic and you'll find this one. You'll also find the last one and tons of other webinars about organic farming and research. We'd also be very grateful if you could fill out the follow-up email survey, which you'll be receiving later today. So we do have a couple of questions coming in, and I just wanted to encourage any of you to ask questions. We had one question, I believe it was for Stephen, about the biochar grant. Would you like to tell us a little bit more about that?
Yeah, sure. We have a local listserv that advertises things coming up, a young farmer coalition listserv in the Hudson Valley. And one day there was it's called Four Corners Carbon, is the kind of the program, and they're working with sourcing from a place in Manhattan called New York Carbon. So it's a byproduct of the lumber industry, and it's particularly white pine biochar. And I've been doing a little bit of research on biochar. I have not implemented on the farm yet. It felt like and in my research, it was all about basically that biochar is not a fertilizer. It's an amendment that once inoculated with compost can become a very long lasting soil additive that allows the microbiome to live within it. So I don't know a lot of detail about these particular companies, but that is how I received this grant. And I'm still learning about biochar. The amount of times that things need to be inoculated, I've gotten some varying information. What I'm planning on doing with it is applying it to a field where our alliums are going to go, incorporating it and growing a cover, and then applying compost about a month before I plant. And so these alliums will be overwintering in this. It'll be doing some field inoculation with the microbiome that's already there. And then I'm going to hit it with a shot of additional compost upon planting and hopefully reap those benefits next year. It's also a part of the farm that we don't irrigate. It's just too much of it's like 8 500-foot rows. And so what biochar can do is, improve soil structure, but also help retain water. So there was some wisdom to putting it in this particular place. But they're going to do soil samples before and after to see what effects might be happening. And that's through Cornell as well. The person that's going to be doing the soil testing. So it seems to be like another research that's somewhat akin to what we're talking about today. That might be the most verbose answer ever, but that's what I got.
Yeah, no, that was really helpful. Thank you. I had a question also. This one I think is for Ethan. You mentioned, Ethan, a support tool to predict the effects of compost from microbiomes. Is anything like that actually available, or is there any way that people can have their microbiome tested to find out if it's favorable or not, or anything like that? Like, is there any way right now that growers might be able to find out more about their microbiomes and what they could do with them or how beneficial they are?
That's actually something that we're trying to set up in our lab right now. It's with a grant that Eli's been working on for a while, but that's one of the main reasons that I came into the lab was to set up. We showed a couple little pictures of it, but it's a relatively inexpensive DNA analysis setup so that we can collect samples from farmers and then hopefully use that and publish our methods so that it could be spread around more. Because right now it can take a really long time to get the DNA from a soil processed or cost quite a bit of money. But yeah, we want to do some testing with it with the New York farmers that we've been working with. And then hopefully get that incorporated into some sort of system like the soil health lab at Cornell, where it could just be incorporated with other types of soil testing.
Okay,
Anything that I
great.
missed there, Eli, feel free to hop in.
Got the thumbs up there from Eli. Okay, you can all unmute yourselves. It's okay. You feel free to jump in if you have anything to add anywhere. So, we had a question here from Darian who wanted to know if anyone has knowledge of Dr. James White, I think that's his name, research on seeds carrying soil microbial partners within them and then releasing them at germination. Does that sound familiar? Eli is nodding. Okay.
I think I can jump in on this one to the best of my knowledge and expertise. I think this is a, understood phenomena that seeds are also carrying microbiomes with them inside of the seed. So I think you're definitely right there, and I'm not familiar with Dr. James White, but that doesn't mean anything. That just means that I'm not familiar with that work. And yeah, I think part of what we know and understand about the seed microbiome, that it's also part of how the plant is able to adapt to different types of environmental conditions. And like right now, we're recruiting a new faculty researcher who, potentially will be studying this aspect of the microbiome, which is, how these plants are, how seeds are, carrying the microbiome along with them, and how it's helping them to adapt to where they're being grown, and it helps to improve their fitness under these types of situations. So yeah, I think this is definitely happening. This is something that I think we can kind of detect in our experiments, but it's incredibly difficult for us to parse it out since we're also using the microbiome from the farms. And so getting kind of to the next stage of that in terms of understanding the microbiome that's also found in organic seeds, and how that's helping them adapt to, these different types of environmental conditions is something that we're interested in. It just requires different types of experimental designs. So I'm not sure that I can say much more than that, other than this is definitely a known phenomena. It's probably going on, and it's probably part of how organic farms and organic seeds that are being produced, ultimately how those seeds are able to adapt to their local environmental conditions, for sure.
Okay, this is a long and thoughtful question here from Liam, so I'm just going to read it out loud so that you can think about it as you go, because it's kind of hard to summarize concisely. It says, very interested to hear that where you save seed matters with respect to microbiome and resiliency. It seemed that Eli was sharing that there was a better resilience of crops grown in a similar microbiome as the soil used to produce the seed. I was thinking about this in relation to the evolutionary purpose of seed, which is often for dispersal. Thinking about the coevolution between birds and chili peppers or trichomes on carrot seed, both aids for long-range dispersal. I wonder if the presenters could comment on this potential contradiction. Is there nuance here in terms of the impact of microbiome on resilience for particular genera of plants or even species?
Well,
That's a
I
good
guess I'm
question.
on deck again. Stephen, were you going to jump in on that one or?
Well, that's an interesting thing that I've thought about too, because we get, when we are trying to advertise and talk about regionally adapted seed, we don't want someone to not want our seed that's not in our region. And that is not really in sync with what the Liam had mentioned too about seed dispersal. Generally, we want there to be few borders between those things. So what we focus on is mostly high germ rates and like the ability of that seed to show vigor and selections for vigor. And I do feel at that point, you're kind of on your own with the seed and what you might encounter in that year in your climate. But that doesn't take away from what we have seen is growing the same thing over and over again. We are getting more robust seed. This is why we make selections of our seed every year on the farm as well and continue to plant those. That's really as much as I have to say. I wonder if those things don't have to be a contradiction, but can kind of exist hand in hand.
Yeah.
That's all I got. Yeah.
I love your philosophical take on that. And I think it's also a practical take. Alice, did you want to jump in or was there someone else that wanted to jump in there before I speak again?
I didn't, but
Oh, okay. Sorry, I thought I heard you.
No.
So yeah, Liam, I think that's a really, great and insightful question. And, one of the things that I noticed is that sometimes the seeds do perform better outside of the microbiome where they were produced. And we don't know the specific mechanism for that. Potentially what is going on is that there's kind of a microbiome arms race going on within the soil. And so oftentimes the plant is trying to, defend itself from pathogens and also develop relationships with mutualists. And we don't really know where we are at, various given points within this arms race between the microbiome and the plant. So one idea potentially, and I kind of insinuated this earlier, is that that in certain situations, there might be a release from this kind of microbiome arms race that's going on the farm if the seed is grown elsewhere. It could be that there are some less beneficial associations going on if you do grow a plant year after year within a given microbiome. And so I haven't thought this completely through. I hope this is a safe space to throw around ideas. But I was thinking that, potentially, if you grow a seed outside of its microbiome where it's historically being produced and it performs better, that might be an indication of a release from some sort of pressure that's going on at the farm where it's being produced. That could be a pathogen, for example, or the microbes are taking advantage of that plant and not benefiting its fitness. So kind of extending that out a little bit, if there is a benefit from growing a plant at a more diverse suite of farms, and maybe Stephen and Leah can kind of comment on this, that, perhaps there is benefits to doing rotation of seed production at some larger scale or larger regional scale. But we don't really know. And I think Stephen is right, which is, and we know that crop rotation is beneficial, right? So it could also be that, extending that analogy forward towards seed is maybe also reasonable. And so, I liked Stephen's response that maybe those things are not totally mutually exclusive. And there's a lot of unknowns here. I love the connection with like evolution and dispersal and the reason for dispersal. And I love following that logic of the ecological mechanism all the way through from kind of these deeper historical points of views or evolutionary points of views. And I think there's a lot to unpack there. Stephen and Leah, do you think what are your observations and what do you think about, what's the future of regional seed production? Are the scales that I'm talking about of interest to growers? Do you think it would be beneficial? What are your thoughts? I'm just spitballing here.
I mean,
You want to go first?
I-
Yeah, go ahead.
I'll just jump in. It's sort of a very general thought, and that is, as I do think that regional adaptation is important. Obviously, otherwise we wouldn't really be doing it. But at the same time, if you go out to the big picture, we are all on Earth, right? So there are certain similarities between the soil here and the soil in, California and the soil in, I don't know, Southeast Asia, because we're all dealing with the same planet, right? So even though there are a lot of nuances individually in different areas, and those nuances I think are quite interesting to delve into, there are some generalities too, and the plants that we have, they've become our garden plants because they have been well adapted to a whole variety of these nuances throughout the world, and that's kind of how they've come to us and become real staples of our agriculture and our gardens. So I think that's at least part of the picture.
Yeah, I agree 100%. And I also feel that this is also one of the reasons why we're not really competing with ourselves when we teach others to save seed. We want to offer it to the world and we have a business and we need to make money. But we freely want everyone to know, like, there's processes, there's processes to be taken to get high germinating seeds. So regional adaptation, when you get something, can happen with you in that space once you get it as well. I mean, that's how these stories are told. That Yactine died so fast. I had 200 plants, and then I had about maybe 50 of them survive, and then I spot sowed, and they punched through, and I was able to get a seed crop of lower yield. And I just think there is work that has to happen with seed saving regionally. And so a high germination rate is your best chance for success. And then you can see how it works for you in that space. So we just like to teach everyone the standard practice of how you do this so you can save your own seed and see how that happens for you.
I would also just jump in and say, Stephen, you already know this, but that in the seed growing world, we're pretty lucky, because when we have an absolutely crappy year, crop wise, we have a really good selection year. And so for us, there's, either you're going to have a good production year or you're going to have a really good selection year. And so which it can be frustrating if you're trying to grow a crop for production, but it's also a huge opportunity. And so, there's more upsides than downsides really for us.
Silver lining everywhere.
Great point. Let's see, we have another question here. This is for Eli or Ethan. Are you finding similarities between soils on each type of farm, for example, vegetables to vegetables, orchards to orchards, etc., in regard to microbiome?
Yeah, we have a study that's currently in review out at a journal. And one of the things that we've done is use there's different soil properties maps within the state of New York. And due to some financial limitations, characterizing the soil properties of all of the farms that we've worked with using kind of standardized soil measures was kind of beyond the scope of what we could do. But we've used these soil maps that are generated through geographic information systems to gain some insights into how the soil properties of the farms are potentially linked to the microbiomes of the farms. And there are very strong correlations. So, farms that have similar soil textures, things like the basic properties of soil, like sand, silt, and clay, all of those properties, organic matter, how fragile the soil is. Many of these properties we've collected and through these soil maps, and then we've correlated them to the microbiome of the farms, and they're all highly correlated. So to kind of address the question directly, if you have a similar soil type or similar set of soil properties to another farm, then you might expect that you might have a similar microbiome to that farm. And there's also been some research here, I can post it in the chat in a moment here, that shows that soil microbiomes, the composition of them is highly correlated to soil health metrics, like the soil health metrics that are collected by so the soil health metrics that are collected and then given to farmers from the Cornell Lab of Soil Health. And so these common tests, they basically indicate that they are related to the microbiome, and we might be able to, similar farms that have similar soil health metrics might also have similar microbial properties. So I guess all arrows point towards, yes, similar farms likely do have similar microbiomes. And I think maybe this is a tough point, but I think one of the interesting things about the microbiome is that it can be similar and it can be different too. And even very different microbiomes can have similar functions depending on the conditions. That's one of the things that we found in our research. And so, yeah, it's very difficult to parse out, even if your microbiome is similar to another farm, will you then have similar types of functions? Will your plants still be resilient to drought? Will they also, still be able to defend themselves from pests? And I think that's kind of an open question. And if your microbiome is different from that farm, is that a kind of a good thing or a bad thing in quotes? And I think that's an open question too. And I think some of our research indicates that, and the research of many others, is that there's a lot of what's called functional redundancy in the microbiome. This is very different than other ecological systems, where there are many different species of microbes. They're taxonomically different, and you might have very different microbiomes across farms. But even though you have so much dissimilarity, or your microbiome is so different, this functional redundancy means that these species at different farms, even though they're different, might perform similar roles. So I guess that's a long and complicated answer, which is, yes, we think the soil microbiome is related to these soil properties, but also there's a lot of nuance in there. And I don't know if Ethan wants to jump in or anyone else wants to jump in to kind of follow up on that with anything that I missed. And in the meantime, I'll post this paper in the chat.
I just wanted to, oh, sorry. Okay, go ahead.
I just to add on a short thing. I think oftentimes they're like the microbiomes that can be really distinct but have really similar effects. The effects of microbiomes for plants can be driven by a really small subset of the microbiome, just a few taxa. I mean, if you have one soil that has a super similar community but has a certain fusarium species, that's what does it and has the effect on the plant. So just looking at the whole composition of the microbiome isn't always as telling for the effects on the plants.
Yeah, this is a question that we have that I think is really useful in this discussion here. It's kind of going backward a little bit, but it's an important question. It says from Sasha, when referring to the microbiome of a farm, how much of the farm are we talking about? I've gotten the impression from various sources that the soil microbiome can vary even greatly within one farm, within one crop on a farm. So am I on the right track here, or were you able to generalize a distinct microbiome to each farm?
I can start out on this one a little bit. Just in those 10 farms that we took the samples from the composted and uncomposted plot on the same farm, in that situation, oftentimes the composted and uncomposted plots would differ far more from each other than they would from other farms. We are seeing some consistencies, and a lot of times we'll take samples from like a whole field and then bulk them together before we analyze the microbiome to kind of generalize out some of those differences. But definitely there can be big differences between plots, even on the same farm.
So I guess, yeah, somebody asked a question in the chat about posting some books or other resources on seed saving. And I know Stephen posted a link to John Navazio's book, but if anybody else has any other great suggestions, I know I've always liked Suzanne Ashworth's book, Seed to Seed, and Carol Deppy's book on breeding your own vegetable varieties. And I have a book from Johnny's Seed Company about seed saving. It's a little pamphlet. I don't know if they still publish it, but if anybody else has any other great ideas.
I think the Ashworth book is Seed to Seed, it's called.
Mhm.
That's a really good one. Yeah.
I think we have time for one more question here. We have okay, Carl has been posting some links to seed saving resources and wanted to know if you have any thoughts about seed exchange programs. I know there's some wonderful organizations like the Seed Savers Exchange that conserve and save seeds. I know, though, one issue that sometimes occurs in some of these research projects is that it's very important that when you're sharing seeds with others to make sure that it's disease-free seeds. Um, but does anybody else have any thoughts about seed exchanges?
Nothing beyond what you said about the risk involved in that, but the benefits are amazing. I mean, I know that the seed company years ago was known as the Seed Library in the first years of their operation. I mean, I've been with them for about 11 plus years, but they've existed for 15. And the Seed Library, in its inception, was started in a library in Gardner, New York, and people could literally check out seeds and take them home, plant them, save them, bring them back. And there was just an exchange that happened in the seed company or the seed company was known as the Seed Library, but that library thing became a misnomer. But there were still exchange programs attached to certain art packs that were sold. And yeah, I think it's a great thing. Every now and then something will show up on my desk where it's like, this person produced this seed in this place. What do you want to do? And sometimes that's how we get new things. And we trust the sources, and we have a trials garden for those purposes. So yeah, I think it's a great thing. I think people should do it more often.
I just want to thank all of the presenters, particularly the growers who are here with us today, Leah and Stephen. That was very inspiring to see all your wonderful work and pictures. And thank you, Ethan and Eli, for another webinar. We look forward to any more from your group as your research progresses. So I'd like to just thank you all for joining us today, and thank you for sharing resources in the chat. And we'll look forward to having you on future programs. Thank you.
Thanks, everyone.
Thank you.
Thanks, guys.